National Institute of Biomedical Genomics is known to be the only institute in India devoted with the researches on biomedical genomics, and he is a current professor of ISI Kolkata also. He obtained his bachelor's, master's and doctoral degrees from the Indian Statistical Institute, Kolkata. He did his postdoctoral research at the Center of Demographic and Population Genetics, University of Texas Medical Center, Houston. He has served on the faculty of Department of Biostatistics and Human Genetics of the University of Pittsburgh. He is an elected fellow of all the 3 science academies in India. He has served on the board of directors of the International Genetics Epidemiology Society, IGES, and he was the founding chair of the ELSI committee. He is a member of Human Genome Organization. He is a recipient of also many awards and medals. He is known for the wonderful contributions in mastering of genes and also honored with the title as Gene Guru by medias. I think he requires no more introduction. So I would request Dr. Parthopati Mujumdar to please come on stage, and I would like to request one of our scholars, Devdatta, to facilitate him.
To the IACS for calling upon me to deliver this lecture, and I thank all of you for being present in this lecture. I'm going to talk about genomics and precision medicine. I'm going to specifically talk about cancers, primarily because I work in that field, but genomics is an enabler of precision medicine, not only in cancer but for various other diseases as well. I see that there are children from the schools around here. So what I'm going to do is to define what I mean by these various terms. But essentially what my talk will be is a series of stories, and I'm going to try and relate those stories to both genomics and precision medicine. Let me begin by showing you a picture of these 2 sand dunes. These are 2 sand dunes that look exactly the same. And there is a reason why I'm showing you these 2 sand dunes. The reason being that these 2 sand dunes look similar, but they are actually not homogeneous. If you look at the grains of sand in this particular dune, the grains of sand are very fine. If you look at this sand dune that looks exactly the same as the other sand dune, in this sand dune the grains are very coarse. So even though on the surface of it these 2 sand dunes look exactly the same, the contents of the same or the texture of the sand in the sand dunes are not the same. So similar looking sand dunes may not actually be homogeneous. I'm giving you this analogy primarily because I'm going to talk about tumors, I'm going to talk about cancers, and if you look at tumors, these 2 tumors look very similar. But actually they are not similar in the sense that they are not homogeneous. If you look at the cells in this tumor tissue, these cells look very different from the cells in this particular tumor tissue. This may not become obvious to you unless you look at it under the microscope, but if you do look at it under the microscope, and these are sections, microscopic sections of cells from the tissue, so even though these 2 tumors look exactly alike, at the cellular level, just as the sand dunes at the level of texture of the sand grains are not similar, here at the cellular level, these 2 tumors, the structure of these 2 tumors are quite different. This is what is very important for us because these are tumors that have been taken from a specific kind of cancer, and these 2 tumors may not actually respond to the same kind of treatment primarily because the cellular structure of these two tumors are very different. So what we really want to do is to look at the inhomogeneity or the heterogeneity that there is in these tumors and relate that to the outcome of treatment, and that is what precision medicine is all about. The way that we do it, well, let me first define for you what we mean by genomics. So all of you know that the hereditary information that's contained in the cells of the body are contained in the DNA of each cell. So each cell contains DNA, and the information that's contained in the DNA is called hereditary information, and all of this, the pool of all of this information is called the genome. Okay? The pool of all of the DNA and the information that's contained in that DNA is called the genome. So this is the genome, this is the physical entity, and genomics is the discipline that seeks to understand the entire hereditary information content of an organism. So we have the entire hereditary content of each cell, and what genomics tries to do is to understand the information that's contained in the genome. So that's what genomics is all about. The human genome contains 3 billion nucleotides, 3 billion alphabets that make up the DNA. There is variation among genomes of individuals. So if I take 2 human beings, each one of them will have 3 billion nucleotides, but the sequence of the nucleotides will not be exactly the same. It will mostly be the same, but about 0.1% of the genomes of these 2 individuals will be quite different, and therefore there is variation among genomes of individuals. It is this variation that gives us susceptibility to various diseases, that makes some individuals prone to cancer, some individuals protected from cancer. It is this variation that gives us the difference in our skin color, in our eye color, and so on and so forth. So the reason why we are different, why we look different, we behave differently, we are susceptible to different kinds of diseases is because of the variation that there is in the differences in the genomes that we have. Also, there is variation among genomes of a cell of an individual. So if I sample cells from various parts of the body, when you're born, usually all the cells will look the same, all the cells will have exactly the same amount, same sequence and same nature of genomic information. But as we go through our lives, different cells in different tissues of the body will accumulate different kinds of changes as a result of various kinds of assaults, some of it natural, such as ultraviolet radiation. Some of us will want to become smokers, and smoking has an impact on the DNA. Some people will refrain from smoking, so smokers will have a larger number of changes, and so on and so forth. Usually these changes that are acquired during the life course of an individual, they are called somatic changes, and these somatic changes are not passed on by the parents to their offspring. So the kind of somatic changes that we acquire during our life course, they are not passed on from one generation to another. However, those somatic changes are also responsible for various kinds of diseases, and in fact, cancer mostly is because of those somatic changes, those changes in the DNA that we acquire during the life course, during our own life courses. So there is variation among genomes of a cell, of an individual. So if you sample cells from different parts of the body, if you sample from different cells from different tissues of the body, from my body, and if you sequence these cells, then what you're going to find is that there will be differences in the sequence of these of the DNA in these cells. So there is variation across individuals, there is variation within individuals as well. Cancer, essentially, so I said that I'm going to talk predominantly about cancer. Cancer is essentially a disease of the genome. This doesn't mean that all of these changes that are acquired during the life course of an individual that play a major role in precipitating cancer as a disease will be passed on from parents to offspring. Most of them will actually be not passed on. About 10% of cancers are actually heritable, meaning that they are passed on from one generation to another. 90% of the changes that happen in the genomes of these individuals that are responsible for cancer are actually not passed on to the next generation. So on the one hand, we say that cancer is a disease of the genome, is a genetic disease. On the other hand, what we understand by a genetic disease is that a genetic disease will be passed on from parents to offspring. Cancer is only about 10% of cancers are passed on from parents to offspring, because most of these changes that precipitate cancer are acquired during the lifetime of an individual. If you look at a cancer cells are very disorganized. Usually we have two pairs of chromosomes, we have 23 pairs of chromosomes, and two members of the pair of chromosomes look exactly the same or look very similar. But in a cancer cell, what you're going to find is that bits and pieces of different chromosomes have moved from one chromosome to another. There are some chromosomes, instead of having two copies, there are multiple copies, and so on and so forth. The hallmark of cancer is an unregulated cell growth and unregulated, this kind of major changes that happen in the cells of cancer. So cancer cells are actually very disorganized. So what we need to remember is that cancer is a disease of the genome, but most of these changes that cause cancer are not inherited, are not passed on from parents to offspring. I'm also going to use another phrase called precision medicine, and so let me explain what I mean by precision medicine. By precision medicine, what we want to do is to use genomic information about a person's disease to diagnose or treat the disease, and I'll give you several examples during the course of my talk today. So most of my talk will be presenting to you various examples where we use genomic information to either diagnose a disease or to treat a disease. So essentially what I'm going to do, and I hope to be able to do at the end of my talk, is to convince you that it is useful to use genomic information for the purpose of understanding what kind of a disease it is, and if you get that understanding, to be able to use that information to treat the disease. This is what precision medicine is all about. Today what we do is, irrespective of our genomes, we treat all individuals with that disease with the same drug. Here is a case where we want to tailor-make. We wish to use the tailor-made information on the genomes of these individuals in order to provide treatment in a sort of personalized fashion to each of these individuals. Like I said, one quick thing that we wish to do is to understand the texture, the cellular texture of these two tumors. And as I said, that the cellular texture of these two tumors that look exactly the same may not actually be different. And they are, in other words, the cellular texture is quite different, it's heterogeneous, and this heterogeneity hurts. In what way does it hurt? It hurts in the sense that if you try to treat these two cancers using exactly the same modality, using exactly the same kind of treatment, these two tumors may not respond. So we need to know that this tumor has a different cellular structure than this tumor, and we need to use specific kinds of drugs in order to kill this tumor as opposed to this particular tumor. So this tumor may not even respond to the drug that actually kills the cells in this tumor. And we want to use that information for treatment, and we want to understand that there is heterogeneity even in apparently similar looking tumors. I'm going to start with a disease called lymphoma, and I'll show you how we use genomic information in order to attain our goals, which is to be able to treat patients better. Lymphoma, so there are various kinds of lymph nodes in a person's body, and these are the locations of the various lymph nodes. A lot of it is in the upper half of the body, and there are some in the groin area. So these are called lymph glands or lymph nodes. They produce what are called lymphocytes, and these lymphocytes help defend the body against microorganisms. We constantly expose ourselves to bacteria and other kinds of pathogens and microorganisms, and these lymphocytes help defend the body. These microorganisms are oftentimes dangerous. They cause diseases, various kinds of infectious diseases, and these lymphocytes then are protective factors that are endogenously produced within the body that help us fight these microorganisms. When you have lymphoma, or when a person has lymphoma, the first sign is a painless swelling of the lymph nodes in the neck or under an arm. Normally, when we have these lymph nodes all around, and even if you touch them, if we touch them, we cannot even feel the lymph nodes. When you have a swelling, very often we ourselves can feel that there is a swelling of a lymph gland, there is a swelling of a lymph node. It's usually painless, but that's the first sign of a lymphoma, which is a cancer, which is a disease that can be very dangerous. treatment, once a lymphoma is detected, once it's determined that this is a lymphoma, it's a cancer of the lymph nodes, the treatment is a combination of chemotherapy. They use some chemicals that's injected into the body, and radiation therapy. They usually use various kinds of rays in order to treat lymphoma. If you look at, lymphomas can be of different kinds, but if you look at one particular kind of lymphoma, and I'll not even tell you what this specific variety is, it's called diffuse large B-cell lymphoma. Doesn't matter, don't worry about the name. This is what the cellular structure of that kind of a lymphoma is. But what I'm going to do is to show you what kind of genes express and what is the nature of the expression of genes in this particular cell, in the cells of this particular tissue. So here it is again, let me hold your hands and take you through this. These are names of genes that are expressed in this tissue. So don't worry about these names. Each row corresponds to a gene that is expressed. The green ones, the green labels here are genes that are expressed at a low level. So I'm measuring the quantity of protein produced by that gene. There are genes that are expressed at a low level. That means the protein that they are producing has a low concentration in the tissue, and the red ones have high concentration, so some genes have a high concentration in the tissue. These are the various genes, about 20 genes. Each row corresponds to the expression of this IRF1, for example, in various tissues of the body, and each column then represents an individual. So these are, each row represents a gene, each column represents an individual, and there are about 500 individuals here, all suffering from diffuse large B-cell lymphoma. Now what you see is that normally if every individual, if every cancer patient with respect to these genes expressed in a similar manner, this entire matrix would have been completely homogeneous, would have had the same color, more or less. But what we see is that in a subset of patients, remember each column represents an individual, in a subset of patients, these 5 or 7 genes express at a very high level, and in another set of patients for exactly the same genes, the expression is at a very low level. In other words, not every patient is expressing at the same level for these set of genes, and that happens for these various sets of genes. As you can see that it's green, it's red, green, green. So this set of individuals is expressing a certain set of genes at a very high level, but these sets of individuals are expressing the same genes at a much lower level. Similarly here, these individuals are expressing this set of genes at a low level, while these individuals are expressing the same set of genes at a very high level. The point here that I'm trying to make is that all individuals who are suffering from large B-cell lymphoma are not expressing the genes in a similar manner. What good does it do to find this, get hold of this kind of information? For the one thing that we can see is that just like the two sand dunes, they looked exactly the same, but the structure of the granularity of the sand was quite different. Here also the tumor tissue may look exactly the same, but the genes that they are expressing and the levels of the expression of the various genes are quite different. This means that there is heterogeneity, and with respect to this heterogeneity, then how can you make use of this heterogeneity? I'm not going to get into the details right now. What you can see is that first of all, they have been named differently. So this is called ABC, this is called GCB, doesn't matter what these names are. So these individuals have been grouped into homogeneous classes. So all of these individuals express these set of genes at a high level, this set of genes at a low level, and this set of genes at a moderate level. On the other hand, this set of individuals express these genes at a low level, these genes at a high level. So as you can see, there is a contrast. So these are groups of individuals who have similar kind of lymphoma, but because of the expression levels of the genes, they have been grouped separately. Why do we want to group separately? Because what we also know is that for GCB, for this set of individuals, the 3-year progression-free survival, so you treat an individual for the cancer, whatever treatment you give, you ask the question, how long are they surviving? So if you look at 3-year progression-free survival, meaning that after treatment, the cancer is gone, or more or less gone, and they don't come back and they don't progress, So 75% of individuals who are in this category have a 3-year progression-free survival. So they are free of disease for 3 years, and 75% of them are free of disease for 3 years. While using the same treatment, for this group of individuals, the percentage of individuals who remain free of disease is only 40%. So this means that something is happening, there is some interaction that's going on between the treatment that we are using and the level of expression of the genes, based on which the individuals have been grouped. So it helps to understand what is the nature of expression of these genes, and as a result of that, the treatment modality may now need to be changed, because if you use the same treatment modality, in one case, 75% of patients survive without the disease for 3 years, and in another case, only about half of them survive without cancer for 3 years. So this is what this understanding helps us find new forms of treatment. This is somebody who all of you, I'm absolutely certain, know, and there is a reason why I'm showing her picture. Is there anybody who does not know who this lady is? Her name is Angelina Jolie, and she made headlines a few years ago, and I'm going to describe to you why she made these headlines if you didn't know it. Of course, all of you know it, but so I'll go through this quickly. Let me go through her life history. Angelina's mother, her name is Marcelina Bertrand, died from a relatively young age of 56 from ovarian cancer. She had a faulty gene which she passed on to Angelina. So the gene that causes these kinds of cancers, ovarian cancer in particular, the faulty gene is known, and she passed this gene, her mother passed on the gene to Angelina. And what Angelina then did is that she felt that she was at a high risk of getting this kind of cancer, and she also knew that ovarian cancer and breast cancer are highly linked if you get this faulty gene. So the first thing that she did was to surgically get her breasts removed. This is called mastectomy, the surgical removal of breasts, and she got a double mastectomy done, which is she removed the organ from her body. Now, is that a good thing to do? Once you know that you have a faulty gene that you have inherited from your mother, of course your risk to getting breast cancer is very high, but is the solution removing the organ? We don't know. This is an ethical dilemma. That's her personal body. She can do whatever she wants with her personal body, but obviously this is not the right way of doing things, because if she had a faulty gene that predisposed her to lung cancer, would she be able to remove the lungs? Of course not. Then she would die. All right? So this is called